Jai Hind everyone, I am Pankaj Kumar and today's lecture is about the structure of proteins. And in this lecture, what we are going to cover the Ramachandran plot, alpha helix, beta sheet, and the beta turn. What is the difference between the beta sheet, beta turn, alpha helix, alpha chain, and collagen fiber, fibro, fibroin, myoglobin, and alpha keratin we are going to cover in this lecture. And the most important, which is Ramachandran plot and the CD spectroscopy, we'll cover in this videos. So we'll see the results of the CDI spectroscopy for the different proteins. Most important is that uh, if you didn't watch my previous videos and you want to watch, continue this lecture, and you can click here on the right side link will be shown here for the previous videos, and you can go and solve the questions related to the protein in the description box. Some questions is also there for the carbohydrate, which we already covered in the previous lectures. So you can go and solve that by clicking the link which is given in the description. So let's begin with the today's class. And most important, sometime lectures will be going fast, or sometime lecture will go takes long time to complete. So you can speed up of 1.5x or 1.75x. That will be up to you. So let's see what is there in your lecture today. So these are the topics which we are going to cover in this lecture: introduction, protein stability, uh, secondary structure, and the tertiary structures. So these are the important things. So let's see the introduction. We'll see. So protein is of different structure. Right? In protein, the amino acid will be there. So either it can form a primary structure, and primary structure is what? The primary structure is, so as it is written here, the primary amino acid is linked. It is the structure which is made by the amino acids which are linked with the peptide bond, or in case of the polypeptide it is additionally linked with the disulfide bonds as well. So there is a peptide bond between two amino acids, and there is just a chain of the amino acid which is looks like this, just chain. Or it can be polypeptide, means so many chains will be there, but it will be in the small simple long chain. Now what is the secondary structure? Now the chain which you're seeing here, it will get the proper uh, sequence in the arrangement. The sequence of arrangement of a polypeptide chain, that will called as a secondary structure. So now this amino acid are in the simple one chain. Now what will be happen? It will be follow this type of arrangement. This will follow this type of arrangement. Then it will form a beta sheet. Or what if it will be follow that this type of arrangement? Then it will form a alpha helix. Okay, now the tertiary structure is just the three-dimensional folding of the polypeptide, like beta and alpha, or the two beta or plus alpha. Okay, so like this type of structure, two beta, two alpha, or so many beta only will comes together, and it will form a form a polypeptide by the help of the folding. Then it will called as a tertiary structure. So what is the quaternary structure? Once the two or more polypeptides in, like two or more tertiary structure will come together and form, make arrangement in a space, so it will become like a globular structure. Then it is called quaternary structure. So this is all about the four structures. Now we'll see the protein stability, and more about the introduction. We'll see there is a different type of amino acids, and there are a lot of the proteins which are same in ourselves, but there are a lot of amino acid which are present in you also and present in me also, but it is different. Suppose you can take an example of the like a simple example, you can take an example of a keratin protein, or you can take an example of the hemoglobin. There will be definitely little bit of the change in the sequence of amino acid of your hemoglobin and my hemoglobin. Or you can take a example of the immunoglobulin. So immunoglobulin is also there in you and me as well, but there is a change in the sequence. So that is called as a polymorphic amino acid. So why these sequences are very important? Because if you see, just change in the arrangement will cause a different type of disorders as well.
So you know the example of sickle cell anemia. In sickle cell anemia, what is happening? There is change in the amino acid from the glutamic acid to the valine. So from the glutamic acid, it is forming a valine. It is the cell RBC will become a sickle cell. So just because of the one amino acid change, it become a sickle cell disease. Or you can, if you see the change in the arrangement, like if you delete the particular region of amino acid, or you will delete some amino acid in the protein, then it can cause a Duchenne muscular dystrophy. So it is also type of disorder
in which is occurring because of the change in the arrangement of a amino acid. So this is very important to understand the amino acids.
arrangement are very important for the functioning of a protein. The second thing is that the amino acid are polymorphic. It is not only polymorphic in terms of the two different species or two different individual. It is polymorphic in terms of our body also. Like any dehydrogenase enzyme you can take as an example, which is present in the liver and it may be present in the brain. So the sequence of the brain dehydrogenase enzyme and the sequence of the liver dehydrogenase enzyme will be changed because of their function. So it is polymorphic in terms of not only
Terms of the individual, it is proteins are polymorphic in terms of the uh, species as well, in the same body as well. So this is the introduction. Will see the protein stability. So protein stability is depends on what the protein stability depends on the following things. So, like a covalent bond, it will depend on the covalent bond, or it can depend on the hydrogen bond, or it can depend upon the ionic bond, and it will form van der Waal. It will form van der Waal. It will form a van der Waal forces it will apply. So this is the important thing. Most important, the hydrophobic phobic interaction. Okay. So whenever we'll talk about the protein stability, the most important, the protein will be in their native stage. Any protein which are present in nature, that is considered as a native stage. So one by one we'll see the effect of this all bonds, and we'll see how it is affect the protein stability. So the most strongest one will be the covalent bond, and this covalent bond will be the depend because of the carbon and the sulfur. So sulfur will be there in the cysteine amino acid, and carbon will be there in the all the amino acids. So this will provide the covalent bonds. Now another is the ionic bond, which will because of the polar or polar charge or polar uncharged as well. So usually it will give the ionic bond will come because of the polar amino acids, but polar uncharged amino acid will give you the hydrogen bonds and all. So what does it have? Either the R group or the amino group or the carboxylic group will be having a charges, so it will give the stability. So every time the ionic bond will not give the stability, because sometime it will give the destabilization of the protein as well as. But whenever the protein ionic bond will come, the entropy will increase. Mostly the entropy will increase, and then it can destabilize. Or if the entropy will decrease, the it will help in the stabilization of the protein. So whenever we will see the native stage of the protein, it is means the Gibbs free energy will be zero of that protein. Now hydrogen bond will continue. The hydrogen bond is the weaker bond than the ionic and the covalent bond. Hydrogen bond, it is because of the partial bond which is occur between the solvent and the solute. The partial hydrogen bond which is present in the water and between the solute. So more polar amino acid, it increase the solvation, and charge amino acid interact water and salt. So this is the important thing. What is the solvation? Solvation means there is a layer of the solvent surrounds the solute. That layer is called as a solvation. Whenever the charges will be more hydrogen bond will be formed. As the more hydrogen bond will be formed, there will be more solvation layer will be there. So was it, is it good for the protein stability? So we'll see, as the solvation will increase, there will be increase in the stability of the protein. So strength of the salt bridge increases, it moves to the environment of the lower dielectric constant. What is the dielectric constant, or what is the dielectric constant? So you know, dielectric, there will be, because of the some peptides, some, suppose this is a long peptide chain, and there will be partial positive charge and there will be a negative charge. This will form a dipole. Okay, like that. In the medium, the dielectric constant will be there. So actually, this dipole will be created because of the amino acid, but dielectric constant will be because of material like solvent, how much it can store the electric current, how much it can permeable for the electricity. Okay, so this is dielectric constant. So if you will go lower dielectric constant, the salt bridge will be increases. Okay, if the salt bridge will increase, it means protein stability will be increased. As you can see here, low dielectric constant means low salt bridge, and low salt bridge means high increase in the protein stability. Hydrophobic hydrophobic stability will, because it's a kind of not liking the water, or phobia of the water of the protein. So whenever the phobia of the water will be there, mostly the solvent will be the wa- water only. Then the protein will form a globular structure, and mostly the amino acid which are hydrophobic in nature, they will like go inside the center of the that globular structure. So the hydrophobic protein outer layer of amino acid participate in the hydrogen bond. But if you see the solvation is increasing, the entropy will decrease. So more hydrogen bond of the partial bond will result in the solvation layer, and this solvation layer decrease the entropy, and finally the protein will stable. Easy only. So it's not a confusing, I think. More hydrogen bond will be there, solvation will be there. Now you'll see how hydrogen bond came into the hydrophobic concept. So hydrophobic concept is the, it contain the amino acid like valine, isoleucine, leucine, methionine. These are the hydrophobic amino acid. Now this hydrophobic amino acid will have the partial hydrogen bond, like the periphery molecules amino acid will be there, in which they will participate in the hydrogen bond, and then it will form a solvation. If solvation will be there, it means it is a good for the stability of the protein. So as you can see in the stabilization in the thermophilic organism also, so salt bridge will be at the center, buried in the center of the protein, as in the thermophilic organism. Mostly ionic bonds limit the structural flexibility. And then van der Waals force. The van der Waals force turns into the electric dipole, and it will overall it will give the stability, but not very much, little only. So we seen the covalent bond is the most important. If the covalent bond will be not there, the all complete chain will not form. If chain is forming, there is a partial hydrogen bond, and because of the partial hydrogen bond, it will form a solvation layer, which will increase this stability. Now if it is forming a hydrogen bond, solvation is there, and the amino acid which are not polar, it means they are hydrophobic, they will settle into the center. Now it's done. Now what is there? The ionic, because of the polar amino acids, some and n terminal, c terminal become ionic, and it can destabilize the protein, or it can stabilize the protein as well. And this long peptide chain one end is a positive, one another end is a negative, it completely form a dipole, and that dipole gives the van der Waals force to stabilize the protein as well. So these are the different types of these forces which help in the protein stability. Second topic is what? Second topic is the protein secondary structure. We'll see. These are the amino acid. This will be your N terminal, and this will be your C terminal. And in this, the amino acid will have the alpha carbon, as you can see here, alpha carbon, and then there will be there will be nitrogen, and there will be another carbon. Okay. Now the bond between the alpha carbon and the nitrogen, alpha carbon and the nitrogen, it will be the phi bond. Okay. And the bond between the alpha carbon and this carbon, this will call as psi bond. So I remembered it psi means 2C will be there. Okay, CC means psi, CC psi, and phi I remembered it with the alpha carbon and nitrogen. Is easily you will remember it. There will be one more bond that is called omega bond. What is the omega bond? The bond which you are seeing here, this bond is called as a omega bond. So before continuing this lecture, we have to understand alpha, psi, and omega. Omega is not very much important, but alpha and psi, sorry, I am saying phi and psi bonds are important. Why it is important? Because of this only, it will allow the rotation of the plane. So some it will give the some restriction to the some plane, and it will allow some the movement of the molecule R groups in the different planes. And finally, like it will adjust the one structure in which the protein will be easily set. Now we'll see the secondary structure. In this we'll read the alpha helix we are going to read. Then we'll read the beta conformation. After that we'll read beta turn. And if nothing like any secondary structure which will not lie in the alpha beta or conformation or beta turn, then it will be called as a random coil. So these things we are going to read. Let's start with the alpha helix. So as you can see here, this is the alpha helix, and the helix contain of repeated turns, and this repeated turn will be repeated in 5.4 angstrom. But the Paulings and the Pauling and one more scientist was there. They found it something different. Their calculation was different, but the actual value is 5.4 angstrom for the repetition of these turns. So this is contain 3.6 amino acids in each turn, and if you'll find there is a hydrogen bonding, these are the dash. These dash are the hydrogen bonding between the fourth amino acid and the first nitrogen atom of the first amino acid. So carbonyl group of the first fourth amino acid and the first amino acid's nitrogen atom. So it will form a hydrogen bond between this. So where this alpha helix will found, actually this will found in the alpha keratin, which is a fibrous protein, and it will form the porcupine quills and the regular structure that repeat every 5.4 angstrom. So where it will be found? it will found it will form alpha keratin. We are going to see, and this alpha keratin is a fibrous protein. Fibrous protein. So, what are the factors which will affect this alpha keratin stability or structure? The alanine shows the greatest tendency of to form a alpha helix in most experimental model systems. So you can see there will be if there will be only any negatively charged amino acid will be in the repetition, it cannot stable because it will have a particular one negative charge which will be not going to be stable. So there should be different type of amino acid which should be there in the trends in chain. So important point comes here. The alanine will be nin have greatest tendency to form alpha helix. So the size of the amino acids usually asparagine, serine, threonine, and the cysteine can also destabilize an alpha helix if they are close together in the chain. So this is the very important one. If asparagine, serine, and threonine, if these are close together, then it will destabilize the then this will destabilize the alpha helix if they are close. If these are close, okay. The positive charge amino acid are often found three residue away from the negatively charged amino acid, permitting formation of an ion pair. This is the important point. And the most important point is that the proline and glycine can't form alpha helix. Why? The reason is that the first thing is the proline doesn't have the free amino acid amine group. It doesn't have amine group to make a hydrogen bond. So the important thing is that cannot form hydrogen bond. Okay, cannot form H bond. And why glycine cannot be the part of the alpha helix? The reason is that it can be very flexible. It is very flexible, and that's why it cannot form a. So that's why it cannot form a alpha helix. Another thing is that affect the alpha helix can be affected by the electric dipole. Now we are going to see the beta conformation. Now we are going to see beta conformation. So now this is the time for the beta conformation. If you'll see the beta conformation is just again sequence, a repeated sequence of the amino acid, but not in a proper helical fashion, but it is in the zigzag manner. So this is the zigzag pattern of the amino acids, which is forming, even being in a zigzag form, it is forming a pattern that is called beta conformation. Now the beta conformation, if you'll see, the several strands arranged side by side, all in the beta conformation is called the beta sheet. So this is the same beta strand or beta conformation is both same. Single protein strand in a zigzag manner. This is a beta strand, and that sheet will be the same if the more than one strand is arranged in a side by side. So this is what this is the beta strand. You can see here. This is the beta strand, and it is in the zigzag manner. Now this is the beta sheet, means more than one beta strands are arranged in a side by side. Now it can be as a parallel or it can be in a anti-parallel manner. If you see is in the anti-parallel, their repeat turns, not exactly turn, their turns will be repeated at 7 angstrom. So the repeat periods will be around 7 angstrom in anti-parallel. But if you'll see, the repeat periods in the parallel beta strand is 6.5 angstrom. So what do you think, which will be more stable? Either parallel beta sheet is more stable, or the anti-parallel beta sheet is more stable? So what do you think? Please give me the comment and tell me the reason why you choose the parallel or anti-parallel, which is more present in the nature. So the if your answer is parallel, then it's absolutely not correct, because mostly nature selected the anti-parallel beta sheet in the uh, organisms, because this will have the more spacious 7 angstrom, more repeat sequence are a little far away, so their stability will be more. There will be less restriction. There will be less restriction between the molecules, and they can settle easily. So this is all about the beta strand, and where you will find this beta strand, mostly you will find this beta strand. So you can find in the silk and the hemoglobin, myoglobin. You will find in this beta sheets. Now we'll see what is the beta turn. So till now you've seen there is a one sheet, and there is another sheet. There is another sheet. So there like one, two, three strands are there. Beta strand, one strand, two strand, third strand. So what if there will be turn like this? If this turn will go and attach like this, and again this turn will go and attach like this, or it can attach in anti-parallel manner, like from here to here, and then from here to here. So this turn which I am making with the black line, this is called as a beta turn. That's it. So this beta turn is containing the following thing. This is the beta turn, and it will contain the following most important thing. It is a just 180-degree turn. It is going in this direction. Again, another turn is going in the opposite direction like this. Yes or no? See, this is a 180-degree turn directly, and it involves the four amino acid. It involves four amino acid. Which are these four amino acids are there? So there are the four amino acid which can be anything, but mostly it will contain the glycine and proline. Now this is the important thing. Now it will contain glycine and proline. Often occur in the beta turns. This glycine and proline often occur in the beta turn. The former because it is small, flexible. The glycine is again it is in the beta turn because it is flexible, and the proline is because it will readily assume as the cis configuration. It amenable to the tight turn. It will give the tight turn, which is important for this turn, and it will give the flexibility. Now, the important thing is that they will have the hydrogen bond. They will have the hydrogen bond of the first residue of the forming a hydrogen bond with the amino group of hydrogen of the fourth amino acid. So these two will be there in proline and glycine. Another two can be another amino acids. So remember it, there will be hydrogen bond between the first the first CO, then to fourth hydrogen amino group. They have a hydrogen bond, H bond in this direction. So this is the all about the beta turn. There will be two type of beta turn you can see. So type 1 and type 2 beta is the there, type 1 and type 2. Again there is a no much difference between type 1 and type 2. There is just uh, rotational or conformational changes you can see in the type 1 and type 2. Type 1 is more available in the nature. So this is all about the configuration and the type beta configuration, beta return, and the alpha helix. Now we are moving to the most important topic, which is the Ramachandran plot. So this is the Ramachandran plot. So most important about this Ramachandran plot is that we know there is a dihedral angles between the amino acids, and that dihedral angles will allow only uh, restricted conformation in which the amino acid or the protein can give the conformations, or they can change their R groups in the different planes. So these are the restricted region in which the following amino acid will be found, or the following secondary structure will be found in the given angles. So for the phi and psi degree we are going to see. So psi degrees, I will remind you again, the psi degree is between the alpha carbon and the carbon degree atom. Now you can see, I'll decrease the size of this. Let me. So anti-parallel will be anti-parallel beta sheet, and the parallel beta sheet will be present mostly in the right side upper quadrant, and it will be specifically these dots are representing the anti-parallel beta sheet and the parallel beta sheet. And this marked region is for the right twisted beta sheet. This marked region is the right twisted beta sheet, and this point is for the collagen triple helix. So left-handed alpha helix, which is very rare in the nature, which is present in the left side quadrant, and the right-handed alpha helix will be around 60 degree, minus 60 degree in the right side down quadrant. Okay. So if I will give the first, second, and third, fourth quadrant, so you can remember it with this. So in first quadrant, parallel beta sheet and collagen triplex will be there, and right twisted beta sheet. In second quadrant, left helix alpha helix will be there. Third, right alpha helix, and fourth, nothing will be there. So this is all about the Ramachandran plot, which we have to remember in our mind because questions will be asked from this topic. Now we are going to see the CD spectroscopy result. So in the screen you can see there is a graph between the difference in molar extinction coefficient and wavelength. So we'll see what is the graph it tells. So this graph is telling that if molar extinction coefficient will be around 20, more than 20, the alpha helix will give you the higher peak, and you will find this absorbance of the change in the right and left handed circular polarized lights. So if suppose there is a sample in there will be sample in which you put your alpha and beta proteins. So it will give the lights and the wavelength of the light UV rays, and this UV rays range will be around 190 to 250 nanometer. And if you give this range of the UV rays, there will be left and right polarized lights will be there, and there will be some absorbance will be occurring because of the left and right polarized light, and the difference between the absorbance of the right and left polarized light, it will give this graph, and this peak will tell you the alpha helix is there, in which you will find that is downside it is giving the two peaks, and the beta conformation will be looks like the red line, it is going above the graph, and then it will show the negative graph, and then it will be again comes into the zero. So for the random coil, you can find the random coil. For the random coil, you will see the absorbance will be minus from beginning, and it will be almost zero. So this is all about the uh, spectroscopy, CD spectroscopy result for the alpha and beta conformation. These questions are asked. Just remember it for your exams and for your uh, practicals. So if you have any doubt, you can ask me in the comment. If anybody don't know the molar extension coefficient, you remember it. It is absorbance is equal to, in from the Beer's law, which we already read, is equal to extension coefficient CL. So molar extinction coefficient will be around absorbance in wavelength by concentration per length. So it is a absorbance of unit concentration in unit length of a cuvette will give you the molar extinction coefficient. So thanks for watching. We'll meet in the next video. The video will be longer for the tertiary protein. So we'll cover this tertiary protein. Remember, beta alpha helix you find in the keratin protein, and then beta conformation you will find in the silk hemoglobin. You will find this. So thanks for watching. We'll meet in the next video. Jai Hind, Jai Bhim.